So dear participants, welcome back again. I'm audible to all of you. हेलो।
हाँ जी अभिषेक, हम तैयार बैठे हैं। आपकी स्क्रीन बड़ी हो जाए तो अच्छा है। हेलो,
Am I audible to all of you?
Yeah, you are audible.
Okay, sir. So dear participants, we are starting now the demonstration of LED fluorimeter, which is used to measure the concentration of uranium in aqueous samples. So it is visible on my screen through my camera that the device, the said device LED fluorimeter, and above this device you can see a few chemicals which we have used for making the standards and the buffer solution. So bit by bit now I will be telling all the things about the LED fluorimeter in detail. So measurement of green fluorescence from uranium compounds is a well-known method for measuring uranium concentration in water.
Excuse me, good morning. Abhishek, can the view be enlarged?
Good morning, ma'am. Yes, ma'am, it can be enlarged, ma'am.
Yeah, because it is not clearly visible, as Dr. Mukesh was also asking for the same.
Ma'am, actually, when
you are using web camera for this or mobile camera? Or
no, I'm using the web camera, ma'am. Actually,
web camera, you can take it behind, and then a larger view can be seen.
If we click on that
Yes.
pin, it will automatically enlarge that.
Yeah, that full screen you can also go for that. That will also, yeah, it's okay. But even then, you can also enlarge the area by
actually, while performing the measurements, I will give the zoom.
You have to show the readings. I understand that thing also.
Definitely.
Yes. So okay.
Yes. So shall I start again, ma'am?
Yes, please.
Yeah.
Please
continue. So measurement of green fluorescence from uranium compounds is a known method for measuring uranium concentration in water samples. Earlier, the laser fluorimeter, which was used to measure the uranium concentration, used nitrogen lasers, which emits a pulse of 337 nanometer, and it was a natural choice for exciting fluorescence in early instruments. But a serious problem of that instrument was the limited life of nitrogen laser tube, which was typically a few months. So keeping in view this problem, a new fluorimeter in which the conventional nitrogen laser was replaced with more reliable UV source, LED was used to excite the fluorescence in uranium salt was developed, and that instrument is called LED fluorimeter. So the device which we have in our lab, LED fluorimeter, can measure uranium concentrations from 0.5 microgram per liter to 1000 microgram per liter with a deviation of plus minus 10%. So in this instrument, the readings are achieved by averaging the measurements over 1280 pulses in 1 second. So the instrument is quite very fast to take the measurements. So as you can see, the instrument have a full interactive color touch screen which guides the operator, and the instrument can store more than 100,000 measurements, and the recorded measurements can be retrieved later at any time for further processing. So talking about the working principle of this instrument is based on fluorescence of uranium complexes. So an excitation of uranium complexes with UV light of suitable wavelength, the uranium complexes emit green fluorescence or green light, which can be measured by sensitive photomultiplier tube. Since the fluorescence yield is proportional to intensity of excitation source and concentration of the uranium in sample, the measurement of fluorescence will give the information about the uranium concentration in the sample in which we are particularly interested. So now I'm coming to the parts of this instrument. First of all, the cuvette which we use for the measurement. So this is the cuvette which is used to perform whole of the measurements. It is made of pure silica which have very low fluorescence, and then the instrument is yeah. So this is the cuvette chamber where the cuvette is placed and inserted. This is the cuvette chamber where cuvette is placed and inserted. So when this lid is open, the PMT tube is turned off, is automatically on off mode. So when we close the lid, only then the PMT tube is ready for the measurement. So in this LED fluorimeter, a pulsed UV LED is used to excite the fluorescence in uranium complexes, and a sharp cutoff filter transmits only the wavelengths which are shorter than 440 nanometer from the LED. So why we use the pulsed UV LED in this LED fluorimeter? So we know generally the samples we use in this in which we take measurements of uranium in this using this instrument are generally groundwater samples, and the generally groundwater samples contains other impurities also. So most of the organic species present in the natural water fluorescence fluoresce mainly due in blue region when excited by UV light. So this fluorescence can be blocked partially using by optical long pass filters, allowing only the wavelength about 475 nanometer. However, this does not eliminate the fluorescence from organic matter. So to eliminate the fluorescence from organic matter, what we do that under pulsed excitation, the fluorescence from the organic matter lasts only for a few nanoseconds, whereas the uranium complexes emit fluorescence for few of microseconds. So by proper gating of photomultiplier tube, the remaining fluorescence from the organic matter can be completely eliminated. So that's why the excitation source is a pulsed source in our instrument. Then inside this the body of this device there is a detection chamber which houses photomultiplier tube, and it is fitted along with the suitable filters which transmit the fluorescent light but do not transmit the LED light because they has been gated properly by using some filters. So the photomultiplier tube is kept in off mode when LED are on, and it's switched off for 50 microseconds after suitable delay. So the electronics which are housed inside this box are single board computer, different microcontrollers, op-amps, and different electronic components which further converts different analog to signals to digital signals, and using the software the concentration is shown here. So there are some protocols or important things which should be kept in mind before performing the measurements in this instrument. So the cuvette, which is which we use for the measurement purposes, it is not shown here.
No.
Yeah. So the cuvette, this has this is a component which is made of ultra pure silica, and it should be handled very carefully. Any dust particle on the surface or any fingerprint on the walls of this cuvette or any scratch on the surface of this cuvette or any deposition of any material on this cuvette can affect the precision of the instrument. So the cuvette should also be held by the walls, on the top of the walls, not by the side walls. It should be held from the edges and from the top. And the sample should not have any suspended particles So ground water sample or whatever the sample which we want to measure in the sample, it should not have any kind of suspended particles or suspended impurities which can scatter light and give anomalous results. And every sample or suspended sample which is having suspended impurity should be filtered with a filter paper Wattman grade 602, which can remove the particles of sizes 2 micron or above. So if there is any leak or crack in the cuvette, or by some mistake, by human error, some water gets spilled in this chamber, in the cuvette chamber, then some of the uranium will get absorbed by the anodized layer of the inner wall of the chamber. So there is the anodized layer inside this chamber. So some uranium get absorbed there, and this absorption results in increasing the background fluorescence by several times, which affect the accuracy of the instrument. So this can be done. So this can be removed by taking suitable precautions by while doing the measurements. Now we are starting with how do we use this instrument. Okay. So as you can see, now we are starting from the scratch. So whenever we start performing measurement of uranium using this instrument, so this is the kind of view we get in our screen. So we tap on new sample. So whenever we tap on new sample, there comes three modes. This instrument operates in three modes. One is standard mode, standard addition mode. Second one is calibrated fluorescence, and third one is uncalibrated fluorescence. So these three different modes are used in different occasions. So the first one, the standard addition mode, is a self-calibrating mode, and it does not require additional calibration at any time. Hence, it is recommended when there is presence of fluorescence quenching compounds. Because what happens that whenever we use the standard addition mode, it compensates for the quenching of fluorescence due to presence of other compounds in the sample. So when there is presence of some quenching compounds in our sample, this mode is recommended to be used. Second one is calibrated fluorescence mode, in which the instrument is first calibrated by the fluorescence of some node some known standard, and later when the measurements are carried out on the samples on unknown uranium concentration, the instrument uses the calibration factor measured and calculated using the instrument. Third one, the uncalibrated fluorescence of the uranium concentration in the mode displays the fluorescence from the sample, which will be proportional to uranium concentration in the sample, but the concentration of the uranium will not be calculated and displayed in this mode. This mode is generally used when there is large number of samples we have, and we want to know that which of them shows very large fluorescence. So then this mode is used. So we generally prefer to use this calibrated fluorescence mode. So we tap on this calibrated fluorescence mode, and we have two options whether we want to continue with the previous calibration which we have performed while using the instrument previously, or we want to do a new calibration in the fresh mode. So we are going to do the calibration right now. So here now it is asking to insert a cuvette of 6 ml of water. Water means the distilled water, double distilled water with 10% fluorine. This fluorine is the buffer solution which is used while preparing this sample, final sample for the measurements. So we have to add 6 ml of water, double distilled water with 10% of the buffer solution. So this is the first step which we do for the calibration of the instrument, and this first step involves the measurement of the uranium in blank solution. So that if the water which we are using, and if there is any impurity there, it should be minus from that. So now we are adding 6 ml of water.
Hello, can you put the camera in landscape mode?
It will not give a zoom view if we do it in the landscape mode. So right now,
Okay, fine.
yeah. So now we have added the 6 ml of double distilled water in the cuvette, and we are going to add 10%, that is 0.6 ml of buffer solution into it. As I told you that our cuvette should be clean. So before inserting it in the cuvette chamber, we clean it thoroughly from all the sides off, and we make sure that there is no bubble inside this cuvette which alters our fluorescence. From holding from the top of the cuvette, we insert it inside the cuvette chamber and we close the lid. After closing the lid, we tap on the enter option, and it shows us the measurement. So this gives us that the fluorescence of which has been calculated by the instrument with the average, standard, and the coefficient of variance and temperature also. Then we remove the cuvette from the chamber, and in the next step it is asking us to insert a cuvette filled with 6 ml of standard solution with 10% of the buffer solution. Here the concentration of standard is 10 ppb, which we are using. We can use different concentration of the standard, but we are using 10 ppb for our purposes because groundwater generally have low uranium concentrations. Make sure after every measurement you clean the cuvette with the double distilled water. So now this cuvette has again been filled with the 6 ml of standard solution and 0.6 ml buffer solution. So once we have inserted the cuvette in the cuvette chamber, we tap on enter mode, and here we can see there are different measurements which we have been shown here by the instrument, and there is a calibration factor which is obtained. So as we tap on the enter option now, so it has saved the calibration, the date of calibration, the time of calibration, along with the serial number. At the number, at number of times we have calibrated the instrument since it has started working. So now the calibration of the instrument has been performed. Now we are ready to calculate uranium concentration in any sample which is having unknown concentration of uranium. For that purpose, again we need 6 ml of water sample of unknown uranium concentration, and in which we will add 0.6 ml of the buffer solution. So we have added this 6 ml of unknown water sample and 0.6 ml of buffer solution into it. Now placing it in the inside the cuvette chamber. And pressing the enter option, next option. So from here we can see that the instrument is giving itself the measurements of fluorescence and its average, standard deviation and coefficient of variation. Along with it, it is showing the final concentration of uranium which is present in this groundwater sample. So the sample which we have used is of unknown nature. We do not know the concentration of uranium in this particular sample. So by using this instrument, we found that the concentration of uranium in this sample is 9.57717 ppb. So using the same process, we can measure the concentration of uranium in different groundwater samples. So this is all about LED fluorimeter. After LED fluorimeter, there is one more device which Mehra sir had asked me to show it to you. This is our pinhole cup dosimeter. So we can open it from three sides. From the bottom of side, you can see there are holes. This is the entry channel of the gas, of the air into the pinhole cup dosimeter, and you can see there is a filter paper here which is used to and this is the first chamber, and inside this chamber, this chamber is called radon plus thoron compartment, and you can see these are the two slip pins. Inside these two slip pins, our detector LR115 is placed, and when we open it from top of the side, there is another chamber which is called Radon chamber, and again there are two pin slips between these two pin slips. Again, another LR115 detector is placed. So after placing those two detectors, placing these two detectors on the slip pins, the device is ready to be hanged on the roof, through the roof of the rooms, like this, away from the walls and the floor. This is all about LED fluorimeter and a bit about the two compartments of radon and thoron chambers in the pinhole cup dosimeter. If there are any questions, then you may ask, please.
Yes, Abhishek, there is one question from my side.
Yes, ma'am.
That's okay. We take buffer solution in the water sample. We have to estimate the uranium content in which. What about the sampling? How we sample? Because suppose I took a sample and place it in a glass bottle or somewhere plastic bottle, where have to put, for how long it have to put? Because if we collect from different or far away places, then instrument is not that portable at all places.
Ma'am, generally whenever we collect sample, we take care that the sample is taken from the deep ground so that we get actual groundwater. So
Yeah.
while taking it from hand pumps, we take care that the hand pump, we run through the water through the hand pump for 2 to 3 minutes so that we get deep groundwater. And once we collect the sample in some scientific grade containers, so we we acidify it using the nitric acid, and then we seal tight the container and we transport it to our lab. And it is
Okay. And for how long we can keep it without? Suppose measurement is not taken, then how long it can be kept?
Ma'am, it is recommended that it should be the measurement should be completed within the 10 days of collection of samples, and maximum up to 30 days.
Because due to radioactivity that the uranium is showing.
Yeah, right, right.
Okay. But thank you. Okay.
So, dear participants, if you do not have any other questions, so thank you very, very much for joining us for 5 days of this GIAN course. And I will be sharing the feedback form and the quiz with you in the Google Classroom form. So, please.
Hello, Dr. Abhishek.
Yes, sir.
Myself, Tanveer Ahmad Pandit.
Yes, sir.
I have before interaction, a little bit of interaction with you. So, if you remember. So actually, since there are three modes of this detection of uranium via LED fluorimeter.
Yes, yes, sir.
So as you discussed, so it depends upon the whether the samples are groundwater taken or any other source.
Sir, actually, these three modes are independent of that collection of samples. It just we know that in some areas, whenever we collect samples, there is very high concentration of uranium. In some areas, there is very low concentration of uranium in groundwater samples. So these three modes are designed in a way that if you have any doubt regarding that the contamination is more in your area and you have number of samples space along with you, then you can use these three modes to have an idea that which samples of which area of which samples of which area should be treated with utmost priority. So suppose you have collected 200 or 300 samples and you have to you want to make sure in a very short period of time that which sample is having more uranium concentration area. So you can do one thing, you can do rapid scan using the uncalibrated fluorescence mode, so that you get
Yeah.
an idea that this sample is showing high fluorescence. So you can measure that you measure the uranium concentration in that sample using the calibrated fluorescence mode.
So does it mean that if we are aware the range of the contamination, so what will be the range of the contamination in the samples taken? So what is the particular mode we should go here? And if we are unknown about the range of uranium concentration, so what will be the best mode to use?
So the best mode to use is the calibrated standard mode, because it is giving you the results in reference to the known standard.
Yeah.
So this is the best mode, and the other mode which I was telling, uncalibrated fluorescence mode, it just gives you idea about the fluorescence, not the concentration.
Yes, sir.
So it will give you the fluorescence.
So
So you
since
will
my
have-
domain is not totally uranium, so I have done a little. Sometimes it is shown BDL, below detection limit. So what is that actually? If you
So
for this
BDL
knowledge
means the
purpose,
below
you can-
detection limit of the instrument. So for our instrument, the detection limit is minimum detection limit is 0.5 ppb. So
Yeah.
if your water sample is having concentration below that, the instrument will not show any result. It will show either negative value. So
Yes,
that
sir.
means that the uranium concentration in your sample is below the detection limit of the
Detection
instrument.
limit of the
That's why we
instrument.
write BDL.
Thank you.
Okay. So
Thank you, Abhishek.
So dear participants, if you have any other question, you may ask, please. Otherwise, we can conclude this session. So dear participants, if you do not have any question, so I conclude this last session of this last day of this GIAN course. And thank you very much for your active engagement listening. So I will be sharing the feedback forms and the quiz along with you on the Google Classroom. So please fill up the feedback form and revert it on the same platform, that is Google Classroom. And thank you very, very much for your participation in this GIAN course.
Thank you, sir. Thank you.